Well, phagocytosis can be performed by both unicellular and multicellular organisms. Where unicellular organisms perform phagocytosis, but instead of destroying infected cells or viruses, they use it to eat, for example amoeba. While multicellular organisms use phagocytosis as an immune response, where these cells either completely kill the invading pathogen or present it as antigen to generate immune response against it. We know very well that immune system is an important system of our body that provides protection against invading microbes by innate or adaptive components. Innate immunity guards our body by first and second line defenses, while protection by adaptive immunity is provided by third line of defense. Protection of our body starts with first line of defense. That are the physical and chemical barriers of the body, like skin, saliva, tears, stomach acid, and mucosal lining, etc. Those microbes that have breached first line of defense are then encountered by second line of defense, where group of cells, tissues, and organs work together to provide non-specific or generalized protection, without targeting the specific invaders. Among many other components of second line of defense, phagocytosis is one of the important processes, as it captures or engulfs the invading microbe and then kill it or present it as antigen, so that it can be detected by immune cells involved in third line of defense, that are mainly T and B cells that target specific antigen. There are many cells of immune system that can undergo phagocytosis. These cells that specifically undergoes phagocytosis are called phagocytic cells or phagocytes. We have discussed about phagocytic cells in our previous videos. But here, before getting in-depth knowledge about phagocytosis, let's briefly summarize all phagocytic cells. Different cells in which phagocytosis can take place are monocytes, macrophages, neutrophils, dendritic cells, B lymphocytes, endothelial cells, reticular cells, RBC, and platelets. Here, endothelial and reticular cells mainly have structural role, as endothelial cells support structure of blood vessels, and reticular cells provide strength to lymphoid organs, but occasionally they can act as phagocytes. Similarly, red blood cells and platelets can also undergo phagocytosis at the time of need, but their actual role is not phagocytosis. They are mainly responsible for blood-related roles. Similarly, B lymphocytes mainly provide defense against specific target microbe by releasing antibodies against that specific antigen. Usually lymphocytes are not phagocytic, but they are capable of taking up distinct microbial pathogens by interacting with different receptors. So we can say that all these cells are purely phagocytic cells. Hence, the main cells that are actually excellent phagocytic cells are monocytes, macrophages, neutrophils, and dendritic cells. Where neutrophils have a distinctive, recognizable morphology and myeloid origin. They are short-lived, with a half-life of about 6 hours. Approximately 1,011 neutrophils are estimated to be generated every day in the adult human. These cells are often referred to as polymorphonuclear neutrophils, PMNs, because they have nuclei with 2 to 5 lobes. During homeostasis, neutrophils are produced each day, but do not survive more than a few hours. During infection, production of neutrophils is increased. Then they migrate rapidly to sites of infection, where they kill pathogens. But even after killing, they do not survive, and become a part of pus at the site of infection. Next major phagocytes are monocytes and macrophages. Monocytes are also myeloid cells, as they originate from bone marrow. They contain many granules and have a lobular-shaped nucleus. Monocytes phagocytose have bactericidal activity. Inside the blood, they are immature cells, migrating to their site of activity. And when they migrate into tissues, they become mature, and now they are called as macrophages. All macrophage forms have long lifespans and survive in the tissue for months or years. Macrophages have a central role at the dividing line between the innate and specific immune response because of their role in antigen processing, as part of innate immunity, and presentation to lymphocytes, thus activating specific adaptive immune response against it. Inside tissues, these macrophages take on a number of specialized forms that are when associated with particular organ called fixed macrophages, like Kupffer cells. These are macrophages associated with liver. Alveolar macrophages. As name shows, these macrophages are present in alveoli and contribute to the lungs' innate defenses. Glial cells, that are long-lived macrophages resident in the nervous system. They are involved in clearing dead neuronal cells. Osteoclasts. These are the most specialized macrophages of bone that participates in regulating calcium metabolism. Among phagocytes, lastly comes dendritic cells. In comparison to macrophages, dendritic cells are weakly phagocytic cells, often called as Langerhans cell of the epidermis. But it's not present only in skin epidermis. Somewhat similar cells are also present in other tissues as well. They migrate through the lymphatic vessels or blood to lymph nodes and spleen, where they act as antigen-presenting cells and cause stimulation of T cells. There are separate follicular dendritic cells for presenting antigen to B cells as well, that are specialized in trapping antigen-antibody complexes. So we can say numerous cells are able to ingest foreign materials, but the ability to increase this activity in response to opsonization, so as to acquire antigen specificity, is restricted to cells of the myeloid series, principally polymorphs, monocytes, and macrophages. These are sometimes termed professional phagocytes. Apart from some variations in their content of lysosomal enzymes, all these cells use essentially similar mechanisms to phagocytose foreign objects. So, this was all about different types of phagocytic cells.